I will follow my presentation as the speaker already mentioned. Title of my presentation is the fingerprint distribution features in the population of the Ajara. Ajara is the part of the Georgia, which is the part of the South Caucasus. I am from Batumi Shota Rustaveli State University, which is one of the big university in the region. This is the authors which take part in preparation of this article. And what about the introduction part of this topic? This is the biomedical sciences. We studied some characteristic of the individualism. What does it mean, the individualism of the personality of a human being has its own biological characteristics. In the modern era, there is a varieties of the methodologies, a lot of kind of the modification of the methodologies, which help us to identify persons' individuality. One of the most popular methodologies is the biometric methodologies, using by the, for example, iris recognition, different kind of the face prominence recognitions, and a lot of type of which automatically identifies an individuals based on his or her physiological or behavior characteristic as well. Some of the technologies are based on the equipment technologies, but what about some characteristics? It's some methods. It's very simple. For example, what about the one of the most popular biometric characteristic is the fingerprint. The fingerprint methods is the one of the ancient methods, and in our modern area, it's are the most common methods among the other biometrical identification methods that have been possibility to us to check personal physiological and morphological characteristics. The fingerprint method globally are used in forensic medicines for the identification of the criminals. How we can identify the personalities based on these methodologies? Each fingerprint is composed of specific papillae and ridges into the finger. Every single person has its individual pattern of the fingerprint, and these individuals are made on the different kind of the ridges, islands, ridges, endings, ridge dots, ridge enclosure, so bifurcation in the fingers. What about the fingerprint formation? Of course, this is the one characteristic, one biological trait, and formation of these biological traits comes from the organogenesis time in prenatal life, especially the formation of the fingerprint relief comes after 13 weeks of the embryonic developmental. This is the fetal life when the fingers and toes are well developed, because the fingers and toes developmental process started the early of the 4 week of organogenesis time, and at the end of the 12-week embryo have well recognizable fingers and toes, and after that is going the formation of the so-called fingerprint, which is the characteristic for the all individuals. In this picture are shown the formation of the fingerprint in the embryonic stage. For example, in the first A picture are shown the primary formation of the fingerprint. Here is shown the first loop formation in the surface of the fingers. This one is the development of this loop. Is shown the complete formation of the fingerprint. I want to tell you that these are so individual characteristic that it's not based on genetical information. Of course, it's are multifactorial traits, which means that not only genetical factors take part for formation of this trait, also non-genetical environmental factors during the early pregnancy is most important for the tissue differentiation process and therefore normal organogenesis process. And this is the reason that persons, especially identical twins, also don't have the similar pattern of the fingerprint. At the organogenesis time, most immediately surrounding the amniotic sac, which contains the amniotic fluid. As I already mentioned, these are the fingerprint formation are as genetically as its non-genetically factors. Which is the non-genetical factors during the embryonic stage? Of course, this is the amniotic cavity, which contains the amniotic fluid, and essentially this amniotic fluid can take part a great role for formation of the fingerprint characteristic for the individuals. Essentially, our fetal cells are floating around inside the water balloons, and much like a balloon, amniotic sac has a thin bendable membrane filled with the pressuring fluid. I told that identical twins also don't have the similar fingerprint patterns, especially if they don't share the extra embryonic layers. What does it mean if they don't share the extra embryonic layers? There are different kinds of the monozygotic twins. According, they are sharing the extra embryonic layer, or they are not sharing this extra embryonic layer, especially amniotic cavity. In some cases, we have the twins which separate, as in this picture are shown that they don't sharing the extra embryonic layer. They have different placenta as well. They have different amniotic cavities, amniotic sac. What about in this picture? In this picture are shown the monozygotic twins, which they have the similar placenta, but they have separated amniotic cavity. This kind of the twins called the monochorionic and diamniotic twins. But what about these cases? These cases twins are the diamniotic and dichorionic. But what about monoamniotic and monochorionic twins? In these cases, twins are sharing both extra embryonic layer. They have the same placenta, and also they have the same amniotic cavities. Why I describe this one? In case of these monozygotic twins, in first and second cases, they don't sharing the extra embryonic layer, means that they don't have the similar environmental factors. But what about it in these cases? In last cases, they have same genetical materials as well. They sharing the similar environmental factors, and maybe in this pyphosis, they have the some high characteristic. They have the similar fingerprint pattern. But what about in first and second cases? They have similar genetical materials, but they don't have the similar environmental factors, and fingerprints in these cases they have similar fingers, and our fingers subjected to the pressure from the fluid around us, and during embryonic life, as we touch the wall of the amniotic sac, the soft developing skins, which is at the 12th week of embryonic development of our fingers, bend and wrinkled, forming the wrinkled, so-called the fracture ridges of the fingers. Prints of the palm of the hands and the bottoms of the feet are also formed in this way. Since no two babies will move around the amniotic sac the exact same ways, not even identical twins have the same fingerprint, as I mentioned. What about the majority type of the fingerprint categories? In this picture are shown three main types of the fingerprint pattern. This is the loop, whorl, and arched. But this is the primary characteristic of the fingerprint. Primary, it's are detected how many type, which categories has the person. After that is identified the secondary characteristic, how many loops is present, how many whorls, number of the loops, whorls, and arched, and also purification, dots, and a lot of other secondary characteristics are also most important for identify the individual characteristics. Based on this primary information, based on these three categories of the fingerprint, in world population, majority are spread the loop fingerprint patterns. 60% of individuals have these categories after is coming the worlds, and only 5% of the individuals have arched type of the fingerprint. What about our research materials? Of course, we have studied as a primary, as a secondary characteristic of the fingerprints, and also we studied the other combination with the correlation fingerprint with other characteristics, other biological characteristic. But now in this conference, I want to tell the only distribution of the fingerprint pattern in our population. Data from 210 individuals have been studied and analyzed in our current research. Most of the studied individuals, 55% we are females, number 116. What about the rest part was in males? We are males, 94%. In our study, all voluntaries which take part in our studies, age range of these voluntaries, we are between 12 and 24 years. Data collection was performed with the participation of the students from Batumi Future School and also students from Batumi Shota Rustaveli State University. How we collected our data. Prior to data collecting, voluntaries we are provided with some proper research information in advance. They were informed of the purposes and objectives of the scientific research and sustained the writing content for the participation in the studies. As I already mentioned, our some voluntaries was lower than 18 age, and in these cases, we have done the content from the parents. Research materials we are collected in the time intervals of the, it are taken from the 2021 year. The research has been done duly approved by the Research Ethics Committee from the Batumi Shota Rustaveli State University. What was the inclusion criteria to the research? For conducting the proper and relief study, we defined some criteria for participation in the current study. The major accent was focused on the high and secondary school students aged 12 and 24 years. The most important criteria for participation in this current result was the undamaged finger relief. The person who belonged so-called the categories to difficult fingers, which has the damaged fingerprint relief, could not participate in the current research. Here is mentioned research materials. The voluntaries involved in the study went through the first phase of this research. This was the interview phase. The interview question, we have done the special interview questionnaire. It was the bilingual questionnaire, Georgian and English. And was informed and was conducted here, names, the contact information, address, detail of the birth, contact information, school faculty courses, assessment of the category, so-called GPA, blood type, ABO, and type of the diets. Of course, we did not mention here in this current the present. I mean, I did not present the today the correlation between fingerprint pattern and the GPI and blood group. It's not analysis at that current time, but we analyze the prevalence of this fingerprint characteristic in our research target group. Research materials for the fingerprint, we choose so-called the pencil techniques, which is quite easy and it's well Also give us possibility to take the fingerprint. We take this fingerprint pattern after analyze the which type of fingerprint it is, and also we can make some calculations after the following. We use these materials for the fingerprint methodology. What about the stages of the fingerprint methodology? First of all, we are drawing the square-shaped paper figure on the special paper and used by the special pencil. This is the 2B pencil, and several times repeating the same process that make the good square. After that, convincing in the cleaning the voluntary's fingerprint fingers. After that, moving fingers several times with this square shaped paper. After remove the fingers, and after removing the fingers, be carefully sticking the adhesive tape on the fingers and fix it. After fixing the fingers, fingerprint, apply the adhesive tape on the sheet. Repeating this process for the all fingers. We take the fingers for the all 10 fingers example, convincing in the good quality images of the fingerprint we obtained. Otherwise, several repetition process we are required until they obtain the results. For this last stage, we study the fingerprint patterns using the magnification glasses. We use the magnification glass, the so-called 3x, by observing and inspecting carefully the outline of each fingerprint and categorize. After that, we categorize the fingerprint accordingly the special categories. This is our fingerprint patterns taken from our voluntaries. What about the results? In this picture are shown the summarization information of about the all 10 fingers of the individuals. I told that we take the 210 persons fingerprint fingerprint, but as I told you, we take our attention all 10 fingers, and all fingers are taken as an individual case. And now we can say that our studies individuals from our studied individuals are taken 2,100 individual cases. And based on all these 10 fingerprints, we identify 4 categories: loops, whorls, arched, and composite. Based on our diagrams here, table here, we can see that chi-square for this statistically is quite high to compare with the critical volume. And also the p-value is the 0.001, and result is significant at the 0.05. Based on our research, we can say that in our population, majority character majority high prevalence has the loop fingerprint type. 58.29% has this characteristic. After is coming the Whorl characteristic, the 691 individual case, which is a 32.9% has whole fingerprint patterns. Arched is appear only in 175 cases, which is the 8.33%. And only 10 cases we have composite type of the fingerprint, which is 0.48 case. As I already mentioned, total are summarized 2,100 individual case. What about the according to the what about the right characteristic, right thumb characteristic? I want to tell you that in some literature are used to identify all 10 fingers pattern, but there's some scientists use the fingerprint individualism patterns detection by using only right thumb fingerprint type. We take this characteristic as well and analyze our result used only right thumb fingerprint patterns. And here is also detect the valuable significance. P-value in this case is also 0.0001, but what about the P is significant at 0.05. What about in these cases? In these cases, the loop and Whorl's individual characteristics are quite not similar, but loop fingerprint patterns distribution is the 49.52%, Whorl is the 45.24%, arched 4.29%, and composite fingerprint is presented in only 2 individual cases in the right thumb. What about the other characteristic which take our attention? We categorize all 10 fingers pattern. Here is mentioned all these 4 type of the fingerprint patterns: loop, whorls, arch, and composite. And in this picture are shown that each number is in the given picture, this 1, 2, 3, 4, 5, and until the 10th, is the name of specific fingers. For example, in leaf, first one is for the right thumb. This is the second is the right index finger. Next is coming the right middle fingers, right ring fingers, right little fingers, and left little fingers, and like so. All fingers are mentioned here, and as you take your attention, in all cases, the prevalence, more prevalent is the loop fingerprint pattern. After is coming the whorl. Next is the arched fingerprint patterns, and less amount of the categories of the fingers for all 10 fingers cases is the composite. And also we analyze our result based on the gender. We are interested to detect the fingerprint type was related to the gender or not. And take our attention here. This was the female information, and this one was the male information. And also here we can see that chi-square is the 10, which is high to the critical volume, which is the 7.8, and the result is the statistical at the significant at the P 0.05. In this picture are shown that loop characteristic is most common in female cases than male. 690% individual cases female have loop pattern of the fingerprint. What about the male? 534. Whorl characteristic is similar, and also what about the arched? Arched 104 cases have females and 71 has males. It's also, if we take our percentages, it looks like as a similar, but we won't tell you that we cannot find quite differences female and male fingerprint characteristic and association with fingerprint and the genders, because in this, if we take this fingerprint type here with the gender based on only one, according to the one type of the some fingerprint patterns, here the result is not significant. Here is also a categorization fingerprint according all 10 fingers, and we can summarize that the fingerprint patterns is not related to the gender. This was our research at that time. As I already told you, we also are trying to analyze the correlation between fingerprint patterns with the ABO blood and the age, because ABO blood and the age is also one identical identification factors for the individualism. And also we are already collected the information about the academic levels of the students, and we try to find the correlation between academic level and the fingerprint patterns. We try to find, because we know that in embryonic life, both of them is the ectodermal origin, and maybe they have the some correlation. There are some correlation between these two individual characteristics. Thank you very much to listen. And I already complete. Thank you.